Welcome back to the cell signaling course. If you recollect, we were discussing a very specific type of signaling which was notch delta type signaling, which represented the cell-to-cell contact mediated signaling. Also, it presented an example where receptor was not having an integral enzymatic activity, neither it associated with any enzyme kind of molecule. It was very unique signaling in terms that multiple rounds of proteolytic cleavage was generating an intracellular molecule which traveled inside the nucleus and carried out transcription, where a repressor complex was activated to an activator complex to mediate transcription. Today we will see a very unique kind of signaling in its own terms, which is integrin signaling, where we will see how the receptor kind of molecule, I would say receptor kind of molecule because here it's a transmembrane protein which has extracellular domain and intracellular domain. Intracellularly they engage with many molecules to mediate a signaling process. Extracellularly they contact the material which is present in extracellular matrix and thus activate the signaling. And we'll also see how together this complex signaling mediated by integrin is affecting the cell motility, movement and growth. So if you think about integrin signaling, it is the receptor or integrins are single pass transmembrane proteins. So in some sense integrin signaling presents a scenario where the receptor as usual is membrane bound and the ligand is somewhere in between. It is not membrane bound and it is not very free, rather it is component of the extracellular matrix. And here you have two subunits which are called alpha and beta subunits. They form heterodimeric assembly for activating the signaling and assembly of the entire signaling complex. They help in the migration, survival, differentiation and support. Particularly some cells, they need to be moving to a different location and differentiate over there. So such differentiation events will be regulated by the integrin kind of signaling. And more often than not, they are responding to the cues which are present in extracellular ligand, which are present in extracellular matrix. Here, mostly the proteins which are fibronectin and laminin is something to which these integrin type of receptors are responding. This presents a unique situation of inside out signaling and outside in signaling. In some sense, it is a representation of bidirectional signaling which we saw with EPH ephrin signaling. So let's have a look at this entire integrin signaling. What are the components, what is the requirement and so on. So as I said, this is a heterodimeric assembly of alpha beta subunit. There are at least 8 alpha subunits and there are 9 beta subunits. They come together in different combination to make alpha beta heterodimer. And the intracellular domain is where you have no enzymatic activity. However, it may associate with many molecules which have enzymatic activity. So in contrast to the notch delta signaling, here the intracellular region is having tremendous ability to interact with so many molecules like we saw for receptor tyrosine kinases. It will interact with effectors which are in the adapter class, which are in the enzyme class and which are basically modulators of signaling and so on. So normally this receptor as a dimer is kept inactive because it requires binding of a protein which is called talin. And this talin protein is also kept inactive before so that the protein talin is also kept inactive so that it is not randomly activating the integrin signaling. So how the signaling is started? One of the molecule which is called ICAP is something when bound to the receptor in the phosphorylated version, and this phosphorylation is carried out by this protein called CAM kinase. It's a serine threonine kinase. So this serine threonine kinase molecule is bound over this serine threonine kinase CAM kinase 2 phosphorylates ICAP, and which induces the affinity between the integrin receptor and ICAP1. Thus the entire thing is kept inactive. This also kind of masks the ability of talin to bind. How this thing will be relieved? Basically, you have the SRC family kinases which phosphorylate this receptor, and here you will have next set of protein tyrosine, phosphotyrosine binding domain containing protein coming and recognizing this phosphorylated receptor and activate next set of molecule, which basically involves removal of ICAP1. Another molecule which is called filamin, that is also bound to the receptor, that needs to be removed, which is held by this molecule called migfilin. So what you are realizing that the presence of a set of molecule in close association with the receptor is keeping the receptor inactive, whether it is ICAP1 or filamin. Their removal is something very critical, and in response to certain growth cues, the SRC family kinases phosphorylate and induce next set of event which will be ensuring the signaling moves forward and receptor is activated. So what generally goes on? I told you for activation you need talin, which is auto-inhibited on the first place. If you consider talin to be a molecule like this, so it is auto-inhibited by acquiring a conformation like this. So this tail which is here is actually holding on to itself and keeping it inactive. For talin activation, there is autoproteolytic cleavage by this enzyme called calpain, which removes the C-terminal tail, and from here it generates talin, which is activated, and this can bind to the receptor. But before that, this alpha beta heterodimeric receptor must also be activated, and this is where we saw a CAM kinase dependent phosphorylation which allowed the removal of the ICAP. Now talin can come into picture here. Talin also has a PIP2 binding domain. So this is representing the PIP2 lipid to which talin is associating and also tethering to the receptor. And this receptor is now active and can engage in multiple different types of signaling involving many proteins. So in some sense, integrin signaling is presenting a kind of circus inside the cytosol where multiple different pathways can merge. They can work together, or they can come and make a complex assembly of signaling cluster. One of the molecule which is importantly shown to be regulating the growth or proliferation kind of activity of these integrin receptors is called RAP1 GTPase. So RAP1, we know it's a small GTPase, and in the GTP bound form they are active, and that is where it will associate with this entire complex of talin, receptor, PIP2, and then activating a separate signaling route. Remember, in case of receptor tyrosine kinases, we saw how RAS was being activated, which was impinging on to the MAP kinase pathway. So even RAP GTPases which are here, they can actually work along with the MAP kinase pathway. One thing which I told you little earlier, that integrin signaling involves inside out and outside in. Basically in represents the cytosol here, out represents the extracellular matrix. Normally for any signaling, we have seen that how signal from outside generates signaling inside the receptor bearing cell. So that is outside in signaling, which basically means that availability of ligand or the agonist and so on is present in the extracellular environment, binding to the extracellular domain of the receptor, activates signaling inside the cell. So that is outside in signaling. We saw in case of EPH-effrin signaling where it was bidirectional signaling, but in both cases it was outside in signaling, because the receptor bearing cell, EPH bearing cell, was signaling from whatever message received outside, binding of effrin ligand and terminating that inside the EPH bearing cell. Similarly, effrin bearing cell was sensing the binding to the receptor in the extracellular environment and sending the signal inside. So both presented the example of outside in, or for that matter I can say that all the signaling which we have seen is more like outside in signaling. In case of integrin signaling, you see inside out signaling. What basically that means, that because of the signaling which started by binding of extracellular domain of integrin to the extracellular matrix molecules like laminin or fibronectin, changes happen inside the cell. Here inside the cell, whatever changes have happened, that is communicated to the extracellular environment, so that migration is made possible, so that motility can be initiated, right? So this is something is called inside out signaling, which is happening in the integrin pathway. So downstream of this activated receptor, which is done by talin binding, PIP2 association and RAP1 GTPase, next set of multiple molecules can be brought over there. Those could be adapters, those could be scaffolds, or enzymes, whether it is a kinase or a phosphatase, or cytoskeleton proteins, they are binders and modulators. For example, in the last slide which we saw, filamin, this is a cytoskeletal binding protein, particularly actin filament binding protein. So you will see how integrin signaling is impinging onto the actin microfilament, its nature, its branching, its dynamics and so on. So once this integrin signaling is activated, the extracellular component, as I said here, the fibronectin or the laminin, in some case if it is a growth factor, if you recollect, there is a receptor in receptor tyrosine kinase which is called fibroblast growth receptor. Fibroblast growth receptor is responding to fibroblast growth factor. Basically, those factors are present into the extracellular matrix. So even such growth factors can also come handy when it becomes when it comes to the activation of integrin signaling. So what has been seen, that often this signaling is anchoring the intracellular actin filament to the membrane with the help of these molecules called vinculin and ARP23. This is a actin binding protein that helps in branching. So basically this is an actin filament, but if ARP23 binds, it induces the branching, which can basically help in structures like philopodia formation or lamellopodia formation, and thus affect the movement or migration. So activated integrins, they will interact with an important component which will help the actin binding protein and eventually affecting the actin dynamics. So if you have read where integrins will be found and what extracellular matrix they will be responding to, so in previous class we discussed about cell-cell junction. One of the cell-cell junction is called hemidesmosomes. where one side is cell, another side is extracellular matrix. So many of these migration related events or motility will be regulated in such hemidesmosomal situation where extracellular matrix is coming in contact with a cell. And in such scenario, the integrin kind of receptor, they are helping it to tether to the intracellular complexes through the actin network, but through the actin filament which is up over there. Now when it comes to signaling and perpetuation of the signal which has been received at the cell surface, so what we know is that this actin dependent anchorage of the cell to the extracellular matrix with help of integrin activation. Here it can involve multiple different types of molecule. I told you if you look at the intracellular compartment near activated integrin receptor, it would look like a circus of things because there is no specific set of activations. We have seen multiple different types of activation downstream of receptor, whether it is directly involving the serine threonine kinases of MAP kinase pathway, or we have seen where cytoskeleton is being perturbed so that cellular movement can happen, or we have seen where GTPases come into picture, as well as you have seen the MAP kinase pathway activation, so on. So it seems that, and in case of we have also seen that in case of receptor serine threonine kinase, how SMAD kind of proteins are activated. In case of integrin signaling, everything comes into the same place. So multiple different types of regulations are there, and like I said in the beginning, the combination of 8 alpha subunits and 9 beta subunits. The permutation and combination which you can think about in these cases is huge. All cells express the alpha beta subunits in various combinations, which helps them respond to the extracellular cues. So beyond this actin and simple assembly of vinculin and R2-3, the alpha beta receptors of integrin family, they further bind to molecule like paxillin, alpha actinin, tensin, FAC. This is called focal adhesion kinase. Basically it gives a signal that how cell is contacting, or where all on its cell surface cell is making contact with the extracellular matrix. You will see FAC kinases getting localized over there, and this FAC is type of tyrosine kinase, non-receptor tyrosine kinase. So you see how CAM kinase came in beginning as a serine threonine kinase. Here we are seeing tyrosine kinase. We will also see phosphatases associating with that. So basically more and more protein come and join this entire thing, and it makes a mega assembly of cluster, signaling cluster, and this signaling cluster will have input from all different types of molecules, whether they are adapter, whether they are scaffold, or they are enzymes. And what will happen over there, that in addition to all these different molecules coming together, it will also activate the MAP kinase pathway via the RTK, which includes the effect on growth, proliferation and survival. You remember one of the first molecule which was coming towards the activation of receptor was SRC family kinase, which was generating the phosphate residue on the tyrosine phosphorylated version on the receptor intracellular domain. This will be recognized by the SH2 domain or PTB domain containing protein. Often PTB domain containing phosphotyrosine binding domain containing proteins recognize this and take the signaling forward towards the growth and proliferation or the MAP kinase pathway. You also saw how RAP1 GTPase was working. That will also impinge onto this growth and proliferation aspect downstream of this integrin signaling. Moreover, since it associates with the actin filaments and allows the branching also, it further works downstream by engaging RAC, Rho and CDC42. These are another set of GTPases. RAC, Rho and CDC42, these are your GTPases. And we know that for GTPases to be activated, you will need at least one enzyme which is in the family of GEF. And when this GTPases have to be inactivated, you will need the enzymes which have this gap activity. So in response to the extracellular environment, signals and so on, it ensures that this different pathways are activated to help regulate the motility, migration, growth, survival and spreading of the cell, because sometimes when cell is undergoing division, it has to detach from substratum and then it has to spread further. So such activities tightly regulated by cytoskeleton, and what we are seeing is that in a scenario where outside environment is extracellular matrix, you will see integrin pathway working on the cells. So this integrin pathway is present in all different cell types. It's not just in case of the cells which are part of hemidesmosomes. This is also present in all different cell types, whether it is immune cell which is moving from one place to another place and responding to the extracellular cues. These receptors in extracellular environment, you can also assess, you can also believe that there will be a lot of metal ions. So these integrins have metal binding properties as well, and thus responding to that. So in combination of alpha beta subunit, the integrins are responding to the components of the extracellular matrix. Often they respond to the proteins which are fibronectin and laminin. Now when we talk about any signaling which is activated, it has to be discontinued as well, and for that, like we saw in case of integrin signaling, the activation started with TALIN inhibition, removal. If I say TALIN inactive, TALIN active, what was happening over there? That there was a protease which was calpain. This calpain actually cleaved autoinhibitory conformation of TALIN with its C terminal being removed and generating the active TALIN. So when it comes to terminating the signaling, what happens that again this calpain comes. Now calpain works on the C terminal intracellular domain of that. If you see all this picture which have been presented before you regarding the integrin signaling, you would notice that so many events are happening where binding of molecule is encouraged to this intracellular C terminal domain. If you proteolytically remove that intracellular C terminal domain, you will not see any of these binding happening. So basically activation of the receptor and downstream signaling and the processes being mediated was anchored at this C terminal intracellular domain of the integrin molecules. Calpain removes this intracellular domain, so you will now have, if you consider this as your active receptor, after calpain digest, you have extracellular entity and this cleaved molecule. This cleaved molecule will be digested and removed from the system, and this receptor also will be recycled so that fresh receptors can be put on the surface. So what we have understood in this class today about the integrins is that they form heterodimeric receptors. We have seen multiple cases of heterodimeric receptors. If you recollect, in case of serine threonine kinases, which where they are part of animal system activating SMAD, or in case of plants, the receptor-like kinases. They were making heterodimeric receptor combination, and their ligand is present in the extracellular matrix. They actually bind to ions like calcium in the extracellular matrix, and the proteins like fibronectin and laminin. The activated integrins, they engage intracellularly with actin cytoskeleton. If you can imagine that cell which is facing the extracellular matrix, it needs to stabilize its position, and that is where cytoskeleton comes into picture, and actin plays very important role over there. Also, when it comes to migrating, moving inside that extracellular matrix, again you will need a dynamic change in the actin cytoskeleton, which will be mediated by integrin signaling. It can engage the soluble tyrosine kinases for signaling. It can engage the GTPases, or further impinge onto the MAP kinase pathway for mediation of this signaling. Importantly, very much like what we saw in case of EPH efferent signaling, the activated receptors were forming a big cluster. So, integrins also form the signaling cluster, and the signaling is completed or is terminated by the proteolytic cleavage of integrins using this protease, which is calpain. The intracellular domain is no more able to hold this cluster or set of molecules near the membrane vicinity, rather it will be degraded because of this proteolytic cleavage. So, we have understood until now very different types of receptor, and in next class we will be able to start with the GPCR, and here we will end with receptors which were more like single pass transmembrane receptor of different types, where receptor ligand, whether ligand was free or ligand was membrane bound, whether they were heterodimeric or homodimeric type of receptor. Thank you.